Okay, are we all here? Who said no? Okay. Good evening, everybody. Okay, come on, it's Christmas. We can do better than that. School is nearly over. Good evening, everybody. That's so much better. Welcome to the Christmas lecture. It is fantastic to see so many of you here. I've got my two very young presenters here. We do believe at Imperial to start science young. So, Sebi, what would you like to say?
Welcome to Imperial College, London.
There's more, there's more. What else? The greatest university in the world.
The greatest university in the world.
Try that again.
The greatest university in the whole wide world.
Yes, he will be enrolling next term, don't worry. We have a fun-packed night for you tonight, lots of science. You've already started off in the senior common room, and that'll be continuing afterwards, after the lecture. Also tonight, we've got the Fringe Festival going on. I don't know if you saw it at the main entrance, but there's even more science to be had out there. So tonight, I'm going to challenge you all, teachers, are you listening? I'm going to challenge every one of you to learn something new tonight. And I want you to take that knowledge and tell other people what you've learned tonight. Science is all about exploration, and science is about having a lot of fun. So have a great time tonight. And now, my other presenter.
Please may you put your hands together for our special guest tonight, Robert Winston.
Thank you. Well, first let me introduce my colleague, Dr.
Oh, hi. I'm here.
So, Sheba basically does all the science in my laboratory. I just talk. And what we're going to do tonight is to talk about reproduction, human reproduction. And that's quite a tricky subject when you've got such a widely ranging audience in age groups, but we'll see how we get on. So what I want to say to you, first of all, is that the extraordinary thing is that every single one of you in this audience, no matter how big you are now, even if you are 70, maybe there are some 70-year-olds here, you were all once invisible. That's quite interesting. And the second thing I'm going to show you is something to do with that photograph behind me, but let's just start with our invisibility. So we're going to do an experiment to start. I want you to rub your hands together really vigorously, all right? Okay. Now, hopefully some of you don't have too sweaty a hand. The drier your hands, the better. Now, we're going to count to 3, and on the count of 3, I want you to clap once as loudly as you can. So, 1. No, no, no. What school do you go to? We'll try again. So, on the count of 3, I want you to all clap simultaneously. So let's go 1, 2, 3. Right, now. Now, breathe a deep breath in, a really deep breath. Now look at the person on your left. Now, each of you have just breathed in the cells from the next door neighbor to you. And that person sitting next to you, each one of those people next to you has got unique DNA. So what you've just done is to inhale somebody else's proteins and their DNA. And quite a lot of this talk this evening is about DNA, that remarkable molecule which makes us each quite unique in a very special way. Unless you're sitting next to an identical twin, you will have breathed in somebody else's DNA. And actually, amazingly, even if you are 20 meters away, you will have got contaminated by somebody else's DNA. Also, their bacteria. That's a nice thought too, isn't it? Right. Now, that photograph on the screen is remarkable because that is a human embryo. It's 2 days after fertilization, and we know it's got about 8 cells, 1, 2, 3, 4, 5, and there are about 3 behind. So it's a perfectly formed human embryo. And to give you an idea of the scale, so you can see just how tiny human cells are, that's balanced on a safety pin, all right? So that is the point of a safety pin. So you get an idea of actually how small we once were. So that across is, roughly speaking, just about under 1/10 of a millimeter. So we are truly, at one stage in our lives, all of us, invisible. And remarkably too, each one of those cells, even though it's in one embryo, each one is capable of becoming a human being on its own. So in purely theoretical terms, if you divided this up into 8 separate cells, you could make 8 identical humans. Not that I want to try and do that, but it is theoretically possible. Now, the other thing I want you to realize, and I think it's quite bizarre this, that I don't know how, Annalisa, how many people are in the room?
About 707.
No, 700 people. Well, what's interesting about that really effectively I could be only talking to about around 150 of you, because the chances of us all being here are about 5 to 1 against. Remarkably, even though that human embryo is perfectly formed, only about 1 in 5 will implant and become a human in the uterus. The rest get lost, and they get lost within about 2 weeks of the menstrual cycle. So what we've done here is to take human embryos and put them back into the uterus at the time when you'd expect the uterus to be able to take the embryo at implantation. And you can see within about 2 weeks, by the time the first period is due, there are only about 20 of those embryos are left out of 100. And actually, roughly speaking, about 18 are going to become people. The rest are simply lost. And because they're so small and invisible, nobody ever notices the fact that they've lost an embryo. It's that tiny. Now, the other thing I want to say to you about this work, and I will be reinforcing that again and again, and Sheba's a very good example, it's very important for you to understand, those of you particularly who are around GCSE level, to understand that actually one of the beauties of this slide is that this is done by Professor Kate Hardy, okay? And she's one of a number of female professors who've worked on my team. Roughly speaking, roughly more than half of my team are female scientists. And I think it's very important to understand that just because you are female, in some reason, you might think you can't compete in science. It's complete nonsense. Women are every bit as good at science as are men, and they should be able to compete equally. And certainly at Imperial College, we're extremely keen to try to promote more and more women doing absolutely international, top-level, cutting-edge science. And again, most of the photographs I'm going to show you have actually been done by the female scientists in my group, including, of course, Sheba Jarvis, who's very kindly agreed to be here today. Now, the interesting thing for me is this extraordinary phenomenon. As a woman ages, her chances of getting pregnant, as I say, about 18 to 25% per cycle. Let's say it's 20% per menstrual cycle. So there are 12 cycles a year, 12 ovulations a year on average. So you can see that the chance of a pregnancy each month gradually falls off. So by the time somebody is in their early 40s, a woman has about a 5% chance per month of getting pregnant. when she's not using contraception. And by the time she's 45, female infertility is really high. So only about 2% of women per month will conceive. So it means that actually a very large number of women in their mid-40s, if they haven't tried to have a baby, may in fact not be able to get pregnant at all because that fertility has so much dramatically declined. Now, there are two messages here. The first message is that it turns out that we are amongst the most infertile animals on the planet, as I'll show you. And the other message is that I think that in our society we're not doing well enough for women, because of course what people are advising women to do is to have their babies early, which is nonsense. What we should be doing is making provision in society for women to have their babies when they need to or when they want to, and providing much better care for children in the workplace, out of the workplace, for women to do work when they can, and so on. And we're not doing that well enough. But the reason why I'm saying that to you is for this reason. It's very important to understand that science is absolutely connected up with society. Science and society run together, which is why I've taken on a chair called Science and Society at Imperial College, because science affects society, and we need to understand science in order to make wise judgments about our society. Even if we never intend to be scientists, it's important to understand science so that we can actually make these decisions. Now, the interesting question about this graph is that there is this declining fertility by early middle age, by 40. And so the interesting question is, does this happen in other species? So what I've done in the next photograph is to photograph one of the oldest living organisms on the entire planet. I'm not referring to my wife. She's just there for scale, all right? And the interesting thing about, I mean, my wife is normal size. She's not a pygmy, by the way. She's a bit shorter than I am, but not much. And she's standing next to this, a monster tree. This is a sequoia tree in Northern California. And the extraordinary thing is that this tree, we know, is about 3,000 years old. And you look round it, and we know that all these saplings are its children, because the DNA has come from that plant. So in plants, that aging process is not anything like as sharp as it is in humans. And it's also true in animals. So here is a very famous ornithologist, George Dunnett. And you see he's holding this bird, which is called a fulmar petrel. It's a seagull, basically, just a posh word for a seagull. And he's holding this seagull, and we know it's the same seagull in both photographs because it's been ringed in 1950. 26 years later, it looks identical. It has the same mate. It's still laying eggs, and those eggs are still completely fertile. So those birds are not aging in the same way as humans. Look at George Dunnett. If you passed him in the street in 1976, having not seen him for 26 years, you probably wouldn't recognize him, because the aging process changes humans. And it turns out that one of the exciting things about embryology, the study of reproduction, is that it gives you an insight into a whole range of other aspects of biological science. So I'm going to show you something I think, personally, it's very interesting, but it's just worth looking back at the person you looked at just a moment ago on your left. Now, even if that person's your best friend, it might be, I don't know, when you have a school reunion in 26 to 36 years, you probably won't recognize them, because we will have changed, we all changed so much. And I remember going to a school reunion of mine some years ago, and I think I recognized about 4% of the people in the room. So it's extraordinary how, as we become adults, we change so much. What is happening is shown by two more female scientists in my laboratory who have published this work. And you can see here is a human embryo, 2 days old, still 2 days old, 2 days old, 2.5 days old, just under 3 days old, 3 days old, nearly 4 days old, and 5 days old. And you'll notice that each one of those tiny cells in the embryo is stained blue, but there are a few cells which are stained red. And that's because these cells are dying. So this process of aging is already happening from the moment of conception. Here you see this process of cell death is a normal process which is happening in your bodies as you sit here listening to me droning on. And in fact, actually, that's very important for you young women, as I'll explain in a second. That cell death process is normal. If you look at your hand, it's got 5 separate digits. But I'm going to show you a photograph of a hand in a mother's uterus a few weeks after conception, and you can't see the fingers at all because they're all joined up together as one piece of flesh. The cell death allows those fingers to actually develop out of the webs. So we all had a webbed hand like a duck or a bird of some kind in the long past when we were first in our mother's womb. Now, let's try and do a little bit of experimental work. So I'm going to need a couple of volunteers of youngish people who like breaking things. So would you like to come up? And we'll have one girl. Do you want to go up the stairs? What's your name?
William Jonas.
Sorry? William. William, okay. And your name?
Tad.
Sorry?
Tad.
Sorry?
Tad.
Chad?
Tad, yeah.
Chad. Okay, so, right. So, I want you to do an incredibly simple experiment. In the human body, one of the largest cells, which is, in fact, only 1/10 of a millimeter across, is the human egg. All right? Can you think of another egg which is a bit bigger?
Chicken egg.
There's an egg from Imperial College for you, all right? And there's an egg from Imperial College for you, all right? Now, go to the edge there, put your toes over the edge, go on, both of you. Now, we've got the camera on that. Right, now then, make sure that you're not going to hit the floor. Right, we're going to count to 3. 1, 2, 3. Drop. Well, you've made a fair mess, haven't you? Now, the extraordinary thing is that there's actually effectively only one cell in that egg. And most of that stuff is yolk and albumin. So what we've got here, and we can put it on camera, is one that we prepared earlier. All right, what's that? Let's show the camera. All right. That's the yolk. What part do you think is going to become the bird? Well, you know what? You can't see the actual single cell there that's going to become the bird. I can't see it. But this is all the food which is needed to feed the developing embryo. Okay? So one of the extraordinary things in embryology is that we need a massive amount of energy to produce a pregnancy. Okay? And that is true in all of these animals. That might be the cell there, actually. That might be just there. Don't know if you can see it, but it's all a bit sticky. Anyway, so basically, that's an unfertilized egg. Okay? Now, Sheba, over to you.
Oh, okay. So, on the theme of eggs.
And we'll have a couple more volunteers up. William,
Yeah.
thank you.
Wow. Okay.
So who wants to handle a human egg? You do, come on. You two.
Okay. A real life human egg.
And what's your name? Kate and Kate and Ari.
Aria.
Aria. Okay. So this is Kate and Aria. And
Hi.
this is not a real human egg, all right? Because what you can see Sheba's done. What have you done to it, Sheba?
So what I've done is I've taken a grapefruit, which is a bit like an egg, of course. And the important thing here is, obviously, it's nothing like an egg. You'll be happy to know. But if I was to take the top off, actually, Aria, you take the top off. It's safe. Ooh, what does it feel like?
It's all
Disgusting.
gooey.
Sticky and gooey. I think you're right. Do you want to have a feel? Oh, ew, I know. So basically, an egg in the human body, although this is not the size of a grapefruit, it's much, much, much smaller, you need a microscope, is coated with this sort of jelly-like substance, isn't it? And it's a really thick gel that covers it called a zona pellucida. Try saying that. Zona pellucida. I can't say it. Anyway, and the important thing here is it's protecting the egg, which is like sort of quiet, basically. And we're going to learn about what happens to it when it's sort of fertilized. And that's a really protective cover. Once that egg gets fertilized, whenever that might be, it becomes really, really stiff. So lots of funny signaling things happen, really, really complex. But basically, this becomes like a shell, and it becomes really, really thick. So I could throw it.
So
I'm not going to do that.
one sperm, which is tiny, less than 100th of the egg in size, is going to come in and is going to hit this egg at some point and enter it. So that zona pellucida is something we're going to see in a second on a film strip. So thank you very much indeed.
You.
Thank you. Now, anybody want any more eggs? Right, now, the eggs, of course, are inside the ovaries in the woman. And so here we've got 4 bits of ovary, which have been looked at in Dr. Anderson's lab in Scotland and my lab in London. So this is a piece of ovary which comes from a little baby that unfortunately died in its mother's womb. This miscarried, but the ovaries were saved for science. So we know that it's packed full of eggs. And here is another one. So that fetus is about 4 months old, and this one is about 5 months old. And then for comparison, you can see a piece of ovary from a 35-year-old woman and somebody in their early 40s. Now, what you can see very clearly, I hope, is that this ovary is absolutely packed full of all these egg cells. And actually, we've made counts, and Dr. Anderson has made counts, and there's somewhere between 3 to 5 million eggs in the ovaries. That's an amazing number of eggs, and theoretically, each one is capable of being completely genetically unique. So that's extraordinary. But as you know, on average, we only have perhaps 2 children in a family, typically, 2.5 children. I don't know if there are any half children here. But basically, there's a massive loss of unique tissue during reproduction. It happens in all animals. It even happens in the relatively infertile human. So here we've got about 5 million eggs in the ovary. 6 weeks later, or 5 weeks later, we've got about 2 million eggs. And by the time a little girl is born, at birth, she will have about 1 million eggs left. And by the time a girl reaches your sort of age, you will have about 300,000 eggs in your ovaries. 300,000, and of course, you're going to lose 1 by ovulation each month. So let's say between the age of 16 to 46 is 30 years. Let's be generous and say 40 years of childbearing life till mid-50s. So 12 ovulations a year, 12 times 40 is just under 500. 480. I hope you can work that out. Anyway, so that's extraordinary. It means that all those eggs are disappearing, because here you can see, here's a woman of 35. She's got 19 eggs in that piece of ovary, and in a similar sized piece of ovary here, we've got just 4 eggs left. So one of the curious things is that whilst you're sitting here wondering how soon you can leave the lecture theatre, listening to droning on and on, you young women are losing about 2 to 3 eggs. And by comparison, some of you young men, anybody over the age of about 10, will have made about 150,000 new sperm in the same time. That's quite extraordinary. So there's a massive difference between egg production, which is not being made virtually at all in women, as far as we know, and sperm production, which is constantly being made, which is one of the reasons why males are often fertile for much longer during their reproductive life. Now, in a minute, Sheba's going to show you sperm, but I think first of all, I think we'll move on with this, shall we?
Yeah.
So let me explain to you. I used to have a teacher who stood in front of a class and say, "What am I?" And what he was, of course, was a womb and 2 tubes, all right? And you'll see that this is my drawing of a womb and 2 tubes, and that is the ovary where the eggs are coming from. So the ovaries are down by my hand. In fact, that would make a perfect ovary, wouldn't it? So I'm holding the ovary there for the moment. Now, the ovary is going to produce the eggs at ovulation. They're going to get into the fallopian tube, and they have to be fertilized somewhere round about where my wrist is. And then if they're fertilized successfully, they will travel up the fallopian tube into the uterus. And each one of you in this room, unless you're an IVF baby, and there may be some IVF babies for all I know in the room, but those of you that were not born as a result of IVF, each one of you spent at least 4 days, about 116 hours, in one of your mother's fallopian tubes, either the left one or the right one. I don't know which one it was, but it's one or the other, okay? Now, what is remarkable, I think, amazing, is that the timing is precise in the human. So it's almost exactly 110 to 116 hours that we spend in the fallopian tube. And what happens is that that's carefully controlled by some more cells. But the reason why we need to show you that is because, meanwhile, whilst the egg is traveling down towards the uterus is being prepared to receive the egg, because there's only one stage of development, about 24 hours, when the fertilized egg will be capable of implanting in the uterus. That'll be round about the middle of the cycle, roughly speaking, about 18 days after the period has started. So what I've done here is to conflate a whole series of scanning electron micrographs. So this is the lining of the human uterus, okay? We've colored it artificially to make it look nicer, and you'll see it developing. Now, what you're looking at is inside the uterus, and a few seconds on screen will represent a lot of hours, and you'll see the moment when implantation is going to be capable. So this lining will develop. These are little pits of glands in the uterus, inside the lining, and you'll see that the lining of the uterus changes as the month develops. So these are red cells, but then you have these lining cells, and they grow little hairs, some of them, like hair, little cilia, and they have other little hair-like projections as well. Now, there's one moment in time, a 24-hour period, when suddenly these cells change and they fill with mucus, rather like the mucus of this egg. And here you are. It feels very similar. So it's very sticky mucus, just like this egg stuff. And then those cells burst, they shed the fluid that they're containing, that nurtures the egg, and then if the egg doesn't implant, the uterus basically loses the lining, it just dies, and there is bleeding from the lining of the uterus, and the next period starts. So that's what happens every month. That's the end of the menstrual cycle. Now, that timing is done by the fallopian tube. And much of my early research was my massive interest in the fallopian tubes. I love the fallopian tubes. They're beautiful. And I'll show you why. First of all, they are extraordinary to look at down the microscope. So here is a fallopian tube. And to give you an idea of scale, from there to there is no more than 2 millimeters across. So it's that narrow, and your egg's going to go down that. And in fact, here is a human egg in the tube, nestling in those folds. So it's got this complicated lining, and that lining has got cells which have those hairs in which beat. And here you can see them. So these are ciliated cells, okay? And the egg travels down between these, and here you see what the egg's eye view will be like. So all these cilia are like seaweed on the bottom of the seabed, and they are all wafting the egg down towards the uterus. Here's a close-up, a high magnification. So here are these cells, and these are the cilia. And what I've done with this film strip is to slow the speed right down so you can see them actually beating. So they're moving. So that's going on in your bodies as you listen to me at the moment. It's amazing, isn't it? And it's going on in you young men as well, because all of us need these cells because we live in a highly polluted atmosphere. If you go down to the Cromwell Road at the bottom of Exhibition Road, we've measured in our lab, in the Wheatrout lab, the level of pollution, the diesel fumes, the particles of diesel, which are quite dangerous to the human body. So one of the things we do to protect ourselves is to have these ciliated cells in our nose, in our trachea, and of course in our bronchi, which attach to the lung. And they're all beating out towards the open air. So basically, when you produce snot, if I can be so impolite, these cells are helping you to get rid of it. They are pushing all that dirty mucus with all the particles into the outside air. And sometimes, if a woman gets some form of infection or inflammation in the fallopian tubes, some of those ciliated cells are lost. And this is a patient who was infertile, and she was infertile because she'd lost so many ciliated cells. And this was a wonderful photograph taken by another female colleague of mine, Gloria Vasquez. So I've shown you photographs now from 4 different female colleagues, and you've met Sheba. I have to think of some males who've contributed to my research. It's going to be tricky to think about it. Now, I want to show you something I think is absolutely stunning, this. I think I'm the only person, this is a real boast, isn't it? I think I may be the only person who's actually filmed human ovulation actually happening. And that's what I'm going to show you now. So what we've done, we've inserted a tiny telescope into the fallopian tube in a live patient who volunteered, and we filmed the inside of the tube, and you'll see the tube, the egg's journey into the tube towards the ovary and away from it. So basically, first of all, we're going to just show you going up towards the ovary. You'll see the ovary, you'll see ovulation, and then I'm going to show you the egg coming down that tube in the reverse direction. So here we go. So here are these folds. And you see, that's the tube there. And we've come to the ovary. And here's the ovary, which looks massive. And there's a tiny little red bleb just on the bottom of it, which is about 1 millimeter across. Here it is. And notice what's happening. The fallopian tube is actually stroking where the ovulation is going to happen. So because the egg is in a sticky mucus, all right, it's going to stick to the fallopian tube. And with a freeze frame, you'll see that mucus, and it's got about 1 million cells in it. There. and there's the egg, and there's about 1 million cells, and they're all being dragged out by the movement of the fallopian tube over the surface of the ovary as it sheds that egg. And then what we've done is to take an egg, stain it blue, so you can see it traveling down the tube between those folds towards the uterus. So that happens every month, whether the egg is fertilized or not. Here's the egg, and there's the helper cells, about 1 million cells around it. So you can just see the darker part there. There's the egg, rather well shown, and there's the 1 million smaller cells which are going to feed it. The chicken doesn't need helper cells because it's got all this yolk that does all the feeding. And there we are. So that is the passage of the egg down the tube. And I think my next photograph is really over to you, Sheba, if you would. Sheba, I tell you what, scientists are peculiar people. They tend to specialize in one area. And I've spent most of my time specializing in the embryo and the fallopian tube. Sheba is an expert on the sperm. So we don't have gender differences in our laboratory in what we're interested in. For some reason, I don't know why, I don't find sperm nearly as interesting as Sheba does. I'm not sure why that is. But anyway, over to you, Sheba.
Okay, thanks, Prof. Okay, so I'm probably going to need some volunteers shortly. But before we do that, hands back down, I'm going to talk you through what are these? You've already had a clue. They're sperm. So what I want to do is be able to show you some moving sperm. And I think, Prof, you can sort of click and go, can't you, with this? Won't they move?
Yeah, they will move.
They'll
yes,
move.
actually.
We'll click and show you.
Yes, one or two of you young lads looked very pleased at the thought that you were producing 150,000 sperm, didn't you? I noticed that, and I want to say something to you before Sheba comes in. Can I do that? Is that all right?
Yes.
Because actually, if you look at these sperm, you think, oh, great, you're really clever. But actually, very few of those sperm are swimming in a straight line. I mean,
It's true.
So that one is quite misshapen. That one's got a funny tail. That one's got 2 heads. And many of them are just dead. So actually, that's one of the puzzles. We males are not nearly as fertile as we think we might be, because although we produce lots and lots of sperm, so many of them are completely incompetent, rather like the rest of us. Sorry, Sheba.
No, it's fine. I think that's a fair description overall, so thank you for that. Okay, so who would like to come up? I'm going to pick maybe 2 volunteers. Okay, you see me? Do you want to come up? And you with the grey top. Let's do it that way. Come on around. Careful. Watch
Please.
the eggs. Definitely watch the eggs. What's your name? Amrol. Amrol. Yeah, okay. What's your name? Elise. Okay, do you want to come on then? No, I won't ask you any questions. I'm going to ask you lots. No, I'm just kidding. Okay, so this is Amrol and Elise, am I right? Yeah? Adam. Okay, completely wrong. So what these 2 are going to do is they're going to help me load up on the microscope so we can all see some live moving cells. Would you like to see that? Yes? Okay. You need gloves. Come along. So this is your science for the night. Pick a glove size. Don gloves on both of your hands. I need 2 of you for this. Do you want to get some gloves, Adam? Great. Okay, so what we're going to do is we're going to, obviously, to look at sperm, we need to look under the microscope, as you can see, because they're very, very small, and there are millions of them. So what we're going to do is we're going to load them up onto a slide. No? Okay, you can have bigger gloves here, if you want. Got one of those. Okay, so we've got a few bits of scientific equipment. This is a bin. This is a pipette. We use this in the lab, and this just helps us load samples in a liquid form. And these are the tips that go onto this. Okay? Guys, are you watching? Oh, yeah. Yeah, good. Alert and orientated, like it. Okay. So what we're going to do is, who wants to hold the pipette? Do you want to do this, Samuel? There you go. All right. Take your time. We're having glove issues.
Right, I want you to look after that egg for me, all right? Would you come
Right,
up
the first
this
person to
front
get gloves on
and
goes.
peel it when I tell
Adam,
you to?
over to you. Hold this.
Yes.
Okay,
Just
I
look
want
after
you
it
to
for
take
the
one of
moment.
these little
What's your name?
pipette tips.
Charlie.
Stick that
Right, so
into
Charlie,
there.
if you just hang on to that egg for me. And
Got it, well done.
either you or
Oh,
your friend peel
okay,
it for
let's
me
do it
on
again.
stage. Well, don't worry, you needn't
Oh,
worry.
you got it. I think that requires a round of applause. That's actually quite hard work.
Will you look after that egg for me? And when I bring you up on stage, what's your name? Jim.
That's what surgeons
James.
have.
James. So James,
And let go. You got something?
the end.
Well,
I'll show
Yeah,
you. Don't
do it
worry.
again.
You don't worry. It's not difficult. Just don't break it, all right, please. Just hang on to it. Give it to your friend then.
And back up.
Just look after it. Please don't break it, all right?
Let me just check. You got
Don't
something?
break
Yeah.
it. Just
Okay. Can I load this on? Well done. Very good. Okay. Put that in the sharps bin. Should we look under the microscope? Yep. Okay, I'm going to put this on, and hopefully we can see we got it projected. Some technical. Oh. Is that on? No. I see him down here. You might need now. Aha, we're going to have some joy, maybe.
turn the light down.
Yeah. Here we go. Are we getting something?
Yeah.
Do you want to look down the microscope, Adam? You did this. Wow. Look down there.
Sheba, whose sperm are they? Who do they belong to?
They don't belong to anyone in particular. Well done. So they're moving quite fast, aren't they? You see they're sort of like trying to wriggle around, find their way around. So ordinarily, in the presence of an egg, what would happen is through that fallopian tube and sort of the womb, the egg sort of is sitting there and the sperm need to find their way to that particular egg. How are they going to do that? Can you think of how that might happen? They've got to go a really long distance. They're really tiny because you had to put them on the microscope and look at them. How do you know? Oh, yeah, Adam, you don't want a question. Errol? Yeah. Okay, over to anyone. How is it going to get to an egg? How's it going to find the egg all that way? Strangely enough, there are some species, like rabbits, for instance, there's a temperature difference. So, for instance, in the womb versus that bit of the branch that Prof was showing you over there, where the egg normally is, there's a temperature difference. So the sperm in sort of rabbits can work out that actually it's a couple of degrees centigrade cooler, so it tries to make its way to a cooler area. So that's pretty clever, these cells, wouldn't you say? Should we give her a round of applause? Very clever cells. But a really, really, really interesting thing scientifically that we've learned in recent times is actually sperm possess a receptor, sort of something sitting on its head, like that it basically helps it work out how to get to the egg. And they're a bit like the receptors you have in your nose when you smell. They're like sort of, they're odorant receptors. So you can sort of waft a particular odorant, and actually these sperm in the lab, I'm not going to show you that today, would waft their way in a really positioned way and get to that egg that way. Isn't that clever? Because we've seen them bumping into each other and being a bit abnormal, to be fair. And actually, once they're in the right conditions, they can find the egg in sort of a linear way, and they sort of go almost like down a highway to the right direction. I think that's all I'm going to say for now. Thank you, guys.
Thanks, guys.
You've done really well. I'll go with the gloves. It's hard, isn't it? Well done. Thanks.
So are you going to capacitate them now?
No, I think I won't.
You won't? Okay. So shall I move on to...
Yeah.
So I'm going to show you something really weird, which I've just recently seen. You know, 'cause, of course, humans give birth to live babies. I mean, we don't lay eggs. One of the things about mammals, with the exception of a very few very primitive mammals, is that we feed our young with milk, we grow hair, and we have, of course, live birth. But actually, some very primitive creatures occasionally can give birth in a live fashion. And if we just, oh, it is working. And these are worms. And these worms, amazingly, are eating their way out of the mother's tummy. Isn't that extraordinary? So these are C. elegans, which are very important in scientific experiments. We use these a lot because, of course, it's not cruel to use them. And those worms always have the same number of cells in their body when they're fully developed. Every worm has an identical number of cells, so they're quite useful to geneticists. So people like myself sometimes use them to study how genes work. One of the most interesting animals, and the one that I actually learnt first at school, was this creature. Does anybody know what that is? It's a sea urchin, absolutely. And the sea urchin's really interesting. I'll tell you why. Because the sea urchin undergoes fertilization using that zona pellucida that Sheba talked about, protecting it. If you think about it, one of the things that was really interesting about that photograph that Sheba showed you, I don't know how many thousands of sperm there were on that screen, but there would be many thousands of sperm in that specimen, and each one of those sperm is genetically unique. Each one, depending on which one hits the egg, each one will make a different human being. That's why, I don't know if there are any brothers and sisters or brothers in the audience, but you don't look like your sibling. You don't look like your brother and sister, because unless you're identical twins, you have different DNA because a different egg was hit by a different sperm, and each egg and each sperm are unique. So that raises a very important problem in biology. Given that there are literally thousands of sperm on that slide and they're all swimming towards the egg, how does the egg ensure that only one sperm gets through and fertilizes it? And that's one of the most interesting things, I think, to learn about. So I'm going to show you how the sea urchin does it now. So here you've got 6 sea urchin eggs, and I'm going to show you the brief moment when the sperm hits each of those eggs. You won't see it happening because it's absolutely massively quick. It's a fraction of a second. But if you look at the periphery, if you look at the round about the egg, you'll see something dramatic happening around the egg which prevents a second sperm getting in. It's called the block to polyspermy, the prevention of a second sperm. So this is what happens. Watch this time-lapse video. It's not even time-lapse, it's real time. So this is actually happening. Right, fertilization's occurred, and immediately the block to polyspermy has developed. There it is, see? Around each of those eggs, there is now a block which prevents a second sperm getting in. Because, of course, if you had 2 sperms in, you couldn't have a viable live sea urchin. It would have too much DNA, it would be abnormal, and it would die. So that's how the sea urchin protects itself. And remarkably, that block to polyspermy occurs in not a very dissimilar way in us. We also have a similar process when we fertilize one of our eggs in our body. I'd like to show you the whole process in the sea urchin because it's so similar to the human, it's almost unbelievable. So this is some film which was taken by a team of scientists in Brazil, in Sao Paulo. So here are the eggs. The animal's mouth is down there, and the eggs are up there. And here are the sperm. And here are the sperm, pretty sluggish. And here's the egg. So just one egg. We're going to see it fertilize. Bang. It's fertilized, and now we've got the block to polyspermy. And all the other sperm around the edge, all unique, can't get in. They're all lost. So they will just die. So reproduction is very wasteful. Now watch this egg. Watch the center of the egg. It's going to divide into 2 cells, 4 cells, 8 cells, and then 16 cells, and then it's going to form a cavity in the middle. There's the cavity with lots of cells all the way around. Now let's see the same process in a human. And this is a film I made I made it in about 1988, I think, so it's quite old. But I'm not sure it would have been filmed before then. This is time-lapse photography, and this is a 4-cell human embryo. And the reason for showing you this particular film, because it is of historical importance, it belonged to a woman who had had a child die of a genetic disease. The child had died at the age of 3.5. So there are a few rare diseases which kill very small children. All of you are very fortunate because you don't have one of these diseases, because they cause the most terrible havoc in families when they occur. And this woman knew she had this disease once the baby had died, and so she wanted to have another pregnancy, but she knew there was always going to be a risk of having another baby that might be damaged. So she asked us if we could find some experimental way of diagnosing whether the embryo that she had was going to be normal or abnormal. So what we did was we devised over a 5-year plan to take away one of the cells to look at the DNA. So that's what I'm going to show you on the film. You remember I said earlier to you that each one of these cells is capable of making a human, so if you remove one, it shouldn't really matter. All the DNA in each of those cells will be identical. So here you see it dividing. This is the human egg dividing, slightly different from the sea urchin, but rather similar. And when it's 8 cells, we're going to take these cells away with a homemade pipette. These are things that we made in a laboratory. There are 2 male scientists and 2 female scientists working on this project. So here is the embryo. We suck it with a tip of a little glass tube, which is so fine you can't see it with the naked eye. And then we drop a tiny bit of acid onto the shell of the egg, the zona pellucida, so we can actually get in where the sperm couldn't get in. So then we can suck out a single cell and we can analyze the DNA. And there you see the cell being taken away. Now the embryo looks pretty ragged, but that's not a problem. That will be perfectly capable of implanting. And we analyzed that particular embryo, showed it was normal, and that week we looked at 3 different women who had fatal genetic disorders in their family, and we put back 2 embryos in 2 of them. The 3rd woman we couldn't treat at that time. Some of these might, there we are, and there's the result. So Lisa and Harriet. So those are the first babies in the world born as a result of diagnosing a genetic disease in an elder brother who had died. And Lisa and Harriet are completely healthy, and they're now 25. So you can see that, well, actually, in fact, Harriet isn't a perfect baby because she's got this blemish on her chin. Okay, that's not because we took a cell away, it's because she fell over about 7 hours before the photograph was due to be taken. She was fine otherwise, she just bumped herself, and she cried a bit, but she was very happy, I think. So they're now adults, and they're now thinking about having their own children. So there is a way of preventing a further generation having that kind of tragedy. And that work is an example of how any of you could work in a team at Imperial College and do that sort of work. You know, I know you might think that Sheba and I are very clever. I can't speak for Sheba, but I can tell you I'm not particularly clever. It's a question really of doing things that interest you and certainly working as a team. And you know what's fascinating? Oh, gosh. You take you two there. You've got around 100 billion nerve cells in your brain. Okay, 100 billion nerve cells. And in each of those cells, there's about 2,000 connections to other cells in the brain. So it's 100 billion times 2,000. So you've got something inside there which is more complicated than any object in the known universe. Okay? Isn't that amazing? It's more complicated than the New York Telephone Exchange, the London Telephone Exchange, the Rome Telephone Exchange, the Moscow Telephone Exchange, and maybe, I don't know, Mexico City's Telephone Exchange together. Now, the reason why that's important is because we think of people as great geniuses, very clever. But you two together can beat any Albert Einstein because you've actually got more brain capacity. So how do we do that? Sheba and I work together. We work as a collaborative team. And all of you are capable of doing that. That's why science is really worth considering. Because that collaboration is actually what makes humans so inventive. It's not being brilliantly clever. It really isn't. It's being interested, being focused, being prepared to work a bit hard to start with. But ultimately, as Annalisa said earlier, science is fun. And I think it's really important to understand that doing something like that, can you imagine how my team of 4 people feel about those babies? They're not their babies, but we feel like their parents. It's something very special that you feel you've contributed. And so you have that opportunity if you do science. And I think that's worth thinking about. Now, I realize the time is getting late, so I'm going to stop fairly soon, but I'm going to show you one more or two more, a couple more photographs. Are you happy, Sheba? You all right with that?
Yeah.
So this is looking down a special thing called an embryoscope, and we're going to look at a human embryo developing, and you'll see, here we're going to see something rather interesting about the human. So basically, it starts off, this is being filmed every 15 minutes, a single shot, and we're filming it for 5 days, and you'll see that at one point, about when it's 3 days old, all those cells come together and form a block of tissue. It's called a morula. There we are, and you'll see the cells are now actually indivisible. You can't separate them. And then after a bit, what's fascinating, they start to separate, and the bit that's going to be the human being is here, and the rest is going to be thrown away. It's the placenta, or put on the rose bushes. It's only that bit that will become the person in the center there. And then it hatches, just like the sea urchin does. So that's quite extraordinary. And this, I think, is truly remarkable. Obviously, we can't film very early development in a human. It would not be legal because we might damage a human embryo. But this, we've been looking at chickens' eggs, and here you see the spine in the chicken. This is the first organ that you can see in the human, the chicken, the mouse. In any animal that's developing, you see what's called the primitive streak, the backbone. Here it is. So the brain is up there, the tail's down there. And here's some unique footage of the baby developing in the human womb. So, here's the primitive streak. And this is the yolk, just like the yolk of the egg that we've been breaking on the floor here. That's the yolk sac. And that's what's feeding this embryo. And we'll see it in about 2 weeks' time in a second on the next photograph. And you'll see, here's the brain. It's still open to the air. There's the spine. And it's a bit more developed now. There's the spine again and the brain, which is a bit bigger. The left side and the right side. Sorry, that's really stupid of me. I pressed the wrong buttons, which is pretty typical. No, it's not going to work now. Oh. So basically, what you're going to see in a minute is the head of the embryo developing, and you'll see something really impressive. You remember I told you that when we're in our mother's womb, the fingers are all fused together. You'll see that. You'll see that this embryo, as it develops, or the human embryo as it develops, has fingers and toes which are fused, just as we'll see in these film shots. Now, there is one hand, no fingers. There are the gills, because early on in development we still have gills like a fish, and the head is down there. There's the gills in close-up. And there's the arm, a little bit more developed in that picture. That's one arm, that's the other arm, with the fingers here, but there are no separation of the fingers, not yet. That'll occur in another week's time. And here you see the fingers starting to separate. So here's the head, the chest, the abdomen, the feet, the legs, and the umbilical cord, which is feeding the embryo in the mother's womb. And then the next stage of development, you see the start of the development of the human eye, which you can see in a bit of film here. I mean, we can go on showing this right through development, but I'm not going to do that because it gets boring. But it's very interesting to see a bit of how the human develops, I think. So here we're seeing the eye developing just here. There. Sorry, there. There's the eye. Right, now, I told you that each of the cells in our body is capable, they're firstly very small, but they're all capable of making all the cells in our body. We have skin cells, we've got brain cells, like I mentioned to you. We have cells in our bowel, in our gut. We have cells which make hair follicles. We have about 220 different cell types. What we can do, and this is work that's going on in Imperial College, this is my colleague Michael Schneider. What he's done is to do something remarkable with 3 embryonic cells taken from a human embryo. Now, about 1/3 of you in this audience, given the current rate of disease, will in old age have heart disease. About 1/3 of us die of heart disease. Look at this. What Michael Schneider's done in his experiment is to make these embryo cells grow into heart cells. So you're watching embryonic cells beating. Now, if you decide to do biology and go to university, that's the kind of experiment you'll be able to continue. We haven't got it very far yet. But look at this. We've made a block of heart muscle here. So imagine being able to replace a diseased heart using embryonic stem cells. That's one of the things of the future, and Imperial College is very advanced in doing that kind of biology. And the other thing we can do is to modify the genes in embryos. So here you see Carol Reedhead injecting DNA into a fertilized egg, and we can change the genes in a mouse, in this case. So there I should put some DNA in, and then that can be then put into the uterus, and we can see what happens with that gene. It turns out that we can change the genes in all sorts of ways. So the last photograph I'm going to show you is the super mouse. The super mouse has been developed by colleagues of mine in Chicago who've made a mouse using modifications of its DNA which can make it run twice as fast and much further than a normal mouse. So what they've done is to modify the muscles, so you've made an Olympic championship mouse. So you're going to see 2 mice racing each other. This is not cruel because they want to do it. They can come off this treadmill anytime they want. So there's the normal mouse, and that's the special mouse. Now, after about 200 meters, that mouse is lazy and it gives up because it doesn't like running that much. So it stops. But this one goes on running. Can it run for an hour, do you think? An hour later, it's still running. Look. Two hours later, it's still running. It can leave the treadmill anytime it wants. 3 hours later, it's still running. And actually, 4 hours later, it's still running as well. So, now, this is really the last picture that I was going to show you, but basically, these are embryos that have been modified, and we now have a technique where we can put DNA in so reliably that in due course, I think by the time you people go to university, it will be possible to modify genes in a remarkable way. And it's a very interesting and a major issue for our society. It might well be possible to make people who can run fast, like those mice, and beat any Olympic record. We might be able to make humans which are much cleverer and have better memory. We'll certainly be able to make humans which are taller. We might be able to make humans who are exclusively blonde, if we really want that. And the issue is, I think, a very important issue, because these are very troubling for society. These are very dangerous consequences of meddling in the wrong way, I think, with the human genes. The question, of course, is, we want to use this to control disease, but is there a risk that somewhere in the world, people might start to meddle with embryos like this to actually try to enhance the human and make a superhuman? So that's a thought for Christmas, really. And that's where I'm going to finish. I did give Charlie and Hannah up there a couple of eggs to peel. If you want to come down, we can do one very quick experiment. Do you want to do that? Yeah, come on then. So. I've lost my matches. I can't do it without matches. Yes, I've got them. Right, so basically, what they're going to do is to peel these eggs for me, all right? Right, Charlie, how are you going to peel that egg? You don't know? Well, I'll tell you how you're going to do it. You're going to go, knock it on the side there. Go on, go on. Keep knocking. You do it the same. Go on. All right, peel them. You see what I did before I started this afternoon. By the way, here's another egg. Right. Now, of course, the egg is surrounded by protein, and it's obviously very heat sensitive. So when we heat up an egg, that protein becomes hard, rather like Sheba showed with the egg over there. So this is an interesting question. Put that egg on top of that decanter. Yeah. How can you get it in without breaking it? No idea? I'll show you. You have to be very quiet now. Here's something you don't learn in school. So what's happened? Well, 1/5 of the atmosphere is oxygen. And what we've done, of course, by the use of fire, we've consumed all the oxygen in the bottle. So we've only got nitrogen left. So we've caused a partial vacuum and that sucks the egg in. Believe me, it's jolly difficult to get the egg out. Thank you very much for listening to me.
Good evening, everybody. I would like to have the fun bit at the end of this and can you help me to say thank you to our speakers? Can you help me say thank you? First of all, everyone, to our helpers who came up tonight. Can you give them an enormous round of applause, please? Secondly, as I'm sure you'll agree, this has been an informative, imaginative, and a really fun presentation. And I'd like both Robert and Sheba to come to the middle so that you can appreciate your support and thanks to them. So can you take the roof down, everyone, please? Thank you. Delighted that here at Imperial College we have people with this enthusiasm buzz to talk to you, the future scientists, the people who are going to take this forward. I would just like to end with a few practical points. After this, we have a reception in our senior common room where the outreach team here will have some presentations and events for you to look at. So once you go out of the hall, there to your right, there'll be some refreshments there as well. And at the main entrance of the college, as Andy said, there's a fringe event if you want to get even more science. So, once again, thank you for coming to our Christmas lecture.
Not too much.